Thank you very much, and I would like to thank Dr. Nagla for her kind invitation, and it's a pleasure to be here again to update you on what we do in my group. I seem to have lost the first slide, which is the title slide, but you have the title in your pamphlets. This is a map of Canada, and this is I just want to introduce briefly the situation with canola in Canada. So these are the provinces that are heavily involved in cultivating canola, and this is where we are located, in Saskatoon, Saskatchewan, and as you can see, the largest producers or the largest farming facilities are in Alberta and Saskatchewan, and to a lesser extent in Manitoba, and to a much lesser extent in Ontario, southern Ontario and Quebec. Here we see more concentration of seed crushers and oil extractors. Canola in Canada, it is a very important crop, primarily because it was developed there, and as you can see, the fractions of the name refer to Canada, oil, and low in erucic acid. Although canola was developed in Canada, Canada is not the largest producer in the world. However, it is the largest exporter of the crop. And depending on the weather conditions from year to year, canola usually competes with wheat for first place crop. And Alan mentioned on Tuesday some of the characteristics of some of these GM crops, where the product that is extracted from the GM crop is not does not contain any GM components. Here is a classical example, and this is why in Canada there is almost a very huge percentage of cultivated canola is transgenic, herbicide resistant, and the reason for the acceptance, one of the reasons for the acceptance is that when the oil is extracted, it leaves behind the transgenic protein in the meal part, so that oil does not contain any transgenic component. So there are three major groups of herbicide-resistant canola that are being produced: Roundup Ready, Liberty Link, and the third one, which is Clearfield, which is produced. The first two are transgenic. The third one is a result of a mutagenesis. Okay, the agriculture mainly depends on water availability. So water is the main ingredient for successful agriculture. If water is disturbed, or the water availability is disturbed, then we see negative results. For example, heat and drought. Heat results in water evaporation, therefore water is not available for the plant to develop and grow. Drought is the same thing, lack of water completely, and often the two occur together, if not separately, but sometimes they they are a consequence of each other. There is also salinity, which affects the osmotic characteristics of plant cells and therefore makes water unavailable efficiently. And there is, as in Canada, there is the freezing problem, which also renders water unavailable through freezing into ice, and this results in either, of course, inavailability of water because it is in a solid form. Furthermore, it damages the membrane and the cell function. So all of these factors affect the crop performance through one key element, and I call it disruptive water status, because if you disrupt water availability, that results in the negative effects on the crop. And it does that through damaging membranes and damaging cells and, of course, the whole organism. So from that point of view, we looked at the membrane. We need to look at the membrane structure. The membrane mainly is composed of a lipid bilayer, which means these are phospholipids. The red circles are the head groups, and you can see the black sticks here represent the fatty acids, which are sandwiched between two layers of the same phospholipid composition. And this is a typical representation of what the phospholipid, what the membrane bilayer looks like. So on the surface where the head groups are, either surfaces, usually these are polar groups, and sandwiched in between them is the non-polar area or part of the bilayer, which is represented by fatty acids. So, oh, there it is. Right. Too late now. Okay. So, at a given temperature, a lipid bilayer can exist in either a solid phase or a liquid crystalline phase. The liquid crystalline phase is what we need to maintain for the cell to function properly, and if it goes into the gel phase, that means trouble. So lipids, all of these lipids have a characteristic temperature at which they transition from one phase to the other, and this is sort of reminiscing of the double-strandedness of DNA. When you raise the temperature, the DNA is called melting, and when you lower the temperature, the DNA is annealing. It's in a sense similar to that is the situation here. So in both phases, the molecules are prevented from, the head groups, the red circles here, are prevented from flip-flopping between the two layers What I mean by that is if we have a look at this green, this is a different head group, it is not allowed to flip to the to the other layer. So that's not allowed. However, in the same layer, the phospholipid can exchange positions with its neighbor very rapidly, and this is what contributes to the dynamic movement of phospholipids in the membrane, within the membrane. So this random process allows lipid to diffuse and wander across the surface of the membrane as the case, as the need might be, and this is what contributes to the so-called cytoskeleton rearrangement. And if we look now at this is the solid phase, which is unfavorable. This process is not allowed. So there is no movement, there is no freedom of movement of any phospholipid within the membrane bilayer. It's important to understand this so that when we design our project, we had to take this into consideration. So this is the key step. This is the key thing that we have to observe. Maintain membrane fluidity. So when we look at the liquid crystalline phase and the gel phase, we see here the degree of unsaturation in the fatty acids is also important, and it plays a very major role in maintaining the mobility of the membrane in what we call as liquid crystalline. And this is primarily because of the presence of double bonds in some of the fatty acids, so that the higher the degree of unsaturation in these fatty acids, the better the chance of getting of maintaining the liquid crystalline phase, because of the configuration around the double bond, it pushes the neighboring fatty acid away, and it creates sort of tunnels or gaps between in the lipid layer, and that would allow for the flow of ions and other species. Here, these are all. This is the other extreme, where all the fatty acids are saturated, and therefore they are stacked. Although they look maybe nicer than this, but they are. The membrane here is very prone to solidifying or getting destroyed or damaged, and consequently compromising the cell. So, so what happens in response to acclimation, and what happens in response to freezing, sudden freezing? In response to acclimation, we see buildup of the unsaturated phospholipids, and as I said, this allows the maintenance of this phase, and also we see a decrease in the saturated molecular species, And here we look at specific phospholipids by names like phosphatidylcholine, phosphatidylethanolamines, lysophosphatidylcholine, and lysophosphatidylethanolamine. These are often contain more percentages of saturated fatty acids. When you, in response to sudden freezing, we see increased hydrolysis of the phospholipids, and this alters the membrane. And there is a massive decline in the levels of phosphatidylcholine, and to a lesser extent phosphatidylethanolamine and phosphatidylglycerol. We also see a dramatic increase in the amounts of phosphatidic acid and lysophosphatidylcholine and lysophosphatidylethanolamine, leading to the adoption of the non-bilayer structure and compromising the membrane integrity and the cellular function. Okay, this is by no means inclusive. This is just the pathways that we have been working with and we have been looking at. It is the major, these are the major signaling points at which phospholipids participate in signal transduction as well as membrane stability. So glucose 6-phosphate is first converted to myoinositol, and this is by the action of myoinositol phosphate synthase. Myoinositol is then by the action of phosphatidylinositol synthase, the first inositol phospholipid is formed, and then this has the option of either being phosphorylated at the 3' position of the inositol moiety to produce phosphatidylinositol 3-phosphate, and that is a component of the membrane and also a signaling molecule. And it is a major participant in maintaining membrane stabilization and fluidity. If PI is phosphorylated differently, here it's either, there are two enzymes involved to form PIP2, either PI4 kinase or PI5 kinase, and if depending on which it is here, the second one comes in here and forms this very important phospholipid, which is then is a substrate for an enzyme for the phosphoinositide specific PLC, phospholipase C. And phospholipase C action on this PIP2 produces two very important second messengers. IP3, which is one of the main, if not the main second messenger that affects calcium signaling in phospholipid signal transduction. This can also be converted to phytic acid, which is in the vegetative tissue. Phytic acid is also a very important signaling molecule. And so, and in addition to these two calcium affecting calcium signal, second messengers affecting calcium signaling, the third one is diacylglycerol, and diacylglycerol also is a second messenger which activates, it has been shown to activate protein kinase C and other protein kinases in mammalian systems, and there are evidence of it being an important messenger in plant systems as well. This is is a very, this is where we have been focusing our effort recently. This pseudo equilibrium or quasi-equilibrium between phosphatidic acid and diacylglycerol, and the reason for this is the presence of diacylglycerol kinase, which converts diacylglycerol to phosphatidic acid, and phosphatidic acid phosphatase, which converts phosphatidic acid back to diacylglycerol. So these two, this is a very important bridging the two pathways of PLC and PLD, because PLD acts on, as well as phosphatidylinositol, it acts mainly on phosphatidylcholine and phosphatidylethanolamine, and in both cases the product is phosphatidic acid plus the head group. So here we see a very interesting bridge situation between the two pathways. So which is responsible for what signal, and what signal is responsible for the traits or the effects on the plants that we study, that we see. Excuse me. So here is a close-up look at the two pathways, the phospholipid lipase C, which produces IP3, which is responsible for activating calcium signaling, and production of diacylglycerol, which also can participate in the synthesis of triacylglycerols, or the oil that we extract. Here, on the other hand, we see phospholipase D, which is converting phospholipids, namely or mainly phosphatidylcholine and phosphatidylethanolamine, to phosphatidic acid and the head group. And we see on this side of the equation the potential for gel phase formation, and on this side of the equation the potential for liquid crystalline phase. This is the favorable side of the equation, and this is we reasoned that we ought to avoid this side of the equation, and therefore what will happen if we disrupt the production of phosphatidic acid? We have done work on PLC, and we are still continuing to do that. And we established certain characteristics, certain outcomes, and we attribute the outcomes to calcium signaling. Here, whatever the outcome that we will see is not really going to be due to calcium signaling, but it's going to be due to other factors. Mainly, this is, as you can see, this is a metabolic reaction leading to a signaling molecule, all right, but it's also reversible, and it is mainly altering the structure of the phospholipid, or the composition of the phospholipid population in the bilayer, the membrane bilayer. So there is no calcium signaling involved in these. So we have an opportunity here of separating the two pathways and looking at the effect of calcium signaling and the effect of this metabolic conversion of the phospholipid. This is an old slide which has been published, and this shows when we started this work, the first phenomenon that we observed was I'll skip these. In this paper we describe the the increase in production of inositol triphosphate under cold and salt stress, which is based on promoting calcium signaling. Now, here this is what I want to describe briefly. We stopped PLD and reduced the amount of phosphatidic acid production, and the way we did this was there are 5 isoforms of PLD. There are 3 major ones in plants: PLD alpha, PLD beta, and PLD gamma. We isolated the gene for PLD alpha, which turned out to have high homology with the PLD beta and gamma. So by anti-sensing using the entire gene, we were able to deactivate all 3 of them. So here we see, this is the enzyme activity, and we can see this is the best line. We have very low PLD alpha activity. PLD beta and gamma on the right also have been reduced in the same line. So with that, we have no doubt that we have disabled all 3 isoforms, and now we go to the testing experiment. Freezing resistance, we see the line, the interesting line here, and this is another derivative line, that it is withstanding freeze treatment, we did this at -5. The experiments are being done at lower degrees, and we see here early flowering by 2 to 3 weeks, as opposed to the PLC plants, which were flowering earlier by a week or so. So, and all these plants are homozygous and advanced generations, and we see consistent inheritable trait, and it has not. In fact, we have even lines that show 1 month early flowering, but we are cautious about that, and we are still investigating that. So this early flowering, in addition to the freeze resistance, and the freeze resistance we tested at the 2-leaf stage, which is a very early seedling, and in order to address the late frost problem in the prairies of Canada, and also at later stages near maturity, in order to avoid early frost, which comes towards the end of the season. So these combined traits, I think, are very phenomenal as far as I'm concerned, combined in one plant by just deactivating one enzyme. Here we see the early flowering, the 3-week early flowering, is not from the perspective of calcium signaling this time. PLC was the calcium signaling. This one is something else, a different mechanism. And this is the days to maturity. The double haploid parent requires 143 days. This transgenic requires 129 days. Also, seed yield was higher. Not much higher, but nevertheless, it is on the positive side. We looked at the oil, there was no change or a slight increase. And the oil composition, we see a decrease in saturated fatty acids, and we see in most of the various lengths of saturated fatty acids. And we see an increase or slight increase in the unsaturated species and the omega-6 and 9. So this tells us that the unsaturation index in these plants have gone up, and therefore explains some of the freeze tolerance that we see. And so at the end, the conclusions are suppression of phosphatidic acid production impacts freeze resistance at the 2-leaf stage, early flowering, and maturation, and enhances seed yield. Suppression of phosphatidic acid production encourages maintenance of the membrane bilayer in the liquid crystalline phase, and multiple isoform suppression of PLD alpha, beta, and gamma proves PLD role in delaying bolting and flowering. And we are continuing to investigate that and verify that. And finally, suppression of phosphatidylcholine hydrolysis raises the unsaturation index and the other species. I would like to acknowledge the funding from Saskatchewan Agricultural Development Fund and my colleagues in my lab, Katrina Nahrina. She is from Russia. She is a brilliant phospholipid person. Wei Yan, a very good, brilliant technician in very good hands in vector construction and transformation. Cheryl Block is my permanent technician, and she's my right arm almost in everything. Thank you.
All right, we are out of time for this morning. Dr. Abdallah has agreed to give her presentation later this afternoon. While we have Dr. Dave up here, let's take one question for him and then we'll work our way backwards through the so that we can make our little coffee time here. So is there one question? Please address that at this time.
Thank you very much. I'm interested in flowering, and this you and Dr. Abdallah found that in 2 different systems that there is initiation of flowering. In your case, there was earliness. Do you think that this is a mechanism that, well, usually plants, what they do, they flower early, escaping the stress? So do you think it's something more related to this in your transgenic plant, subjected to cold tolerance? It's nothing related to trait modified.
Thank you. The fact that we do these tests separately. We don't do freezing and, freezing is done obviously in a separate experiment, but early flowering is a natural occurrence that spontaneously develops in front of us without subjecting the plants to stress at all. And we look at the leaf parameters, the gas exchange parameters, and we find that there are improvements in the stomatal movement and also water retention. And there was no time to mention all this. Improvement in water retention properties and photosynthetic properties as well. So I'm not attributing the early flowering to stress at all. No, there are other mechanisms that we are investigating in this time. Hence, okay. For example, this is one. This is a theory, but we're in the process of verifying it. Number one, we have disabled 3 isoforms of an enzyme. These three, this disablement. Don't forget that PLD leads to phosphatidic acid, which is also a signaling molecule. So you have prevented a signal here. By preventing the signal, the question is, did we affect a suppressor of flowering that is time programmed early on, and hence we see this early flowering? I think the answer could be yes, and if so, then we stand a very good chance of developing some breeding program to suppress that flowering suppressor once we identify it.
All right, looks like we got one more burning question here that we'll take a quick question because we move on.
There has been some breeding research going on, screening for cell membrane thermostability on meat. We found that temperature was about 44 Celsius, differentiating tolerant from susceptible lines. How far we can say that there is a genetic variability among a crop like wheat for cell membrane thermostability from your research?
Without going into the analysis of the membrane structure and the phospholipid composition and the protein composition, I will only be guessing, and I can't give you a guess. I don't know. I'm not familiar with cell membrane structures in wheat, but most likely you have a situation where you are maintaining the membrane on the liquid crystalline phase side.